Hello everyone, welcome back to my channel. I am Nivedita Kauri, MBBS student at Bankura Shomiloni Medical College, West Bengal. Today my topic of discussion is coagulation of blood or clotting of blood. Coagulation is a protective mechanism of our body to prevent excessive bleeding after an injury. It occurs by a cascade of reactions. Why is it called as cascade of reactions? Because there are several factors in our blood. These factors are known as coagulation factors. One activated coagulation factor works on the inactive form of another coagulation factor. Now it becomes activated. This activated coagulation factor then works on another inactive coagulation factor to make it active, and this goes on. So the coagulation factors get serially activated to ultimately produce a clot to stop the bleeding. First, I will talk about the clotting factors or the coagulation factors. There are total 13 coagulation factors present in our blood, of which the sixth is a hypothetical factor. That means it doesn't exist actually. Let us see the name of the coagulation factors. Factor 1 is fibrinogen. Factor 2 is prothrombin. 3. Thromboplastin. Factor 4 calcium ion or Ca2+. Factor 5 is proaxillerin. Factor 6, it's a hypothetical factor. It doesn't exist. Factor 7 is proconvertin. Factor 8 is anti-hemophilic factor A. Factor 9 is anti-hemophilic factor B or the Christmas factor. Factor 10 is the Stewart-Brauer factor. 11 is known as plasma thromboplastin antecedent. Factor 12 is the Hedgeman factor, and 13 is the fibrin stabilizing factor, which is also known as the Lucky-Laurent factor. Deficiency of factor 8 causes hemophilia A, and deficiency of factor 9 is responsible for hemophilia B. Let's move on to the pathways of coagulation. Initially, there are two pathways, the intrinsic pathway and the extrinsic pathway. Factor 10 is activated by both the pathways. Then there is a common pathway to produce a clot. I will describe each of these pathways, but the pathways are little bit complicated, so please pay attention. Let's talk about the intrinsic pathway first. Whenever there is any injury to any blood vessel, the endothelium is injured and the subendothelial collagen is exposed. When blood comes in contact with the subendothelial collagen, the factor 12 gets activated. So, factor 12 gets activated to factor 12A. Here A suggests that the factor is activated. So, factor 12 gets activated on exposure to subendothelial collagen. Factor 12A works on factor 11 to activate it. So, factor 11A is generated. Now, the factor 11A acts on factor 9 and factor 9 is converted to factor 9A. Factor 8 is converted to factor 8A by the action of thrombin, which is generated by the extrinsic pathway. Now, the factor 8A, now the factor 8A, 9A and calcium ion act on factor 10 to produce factor 10A, the activated form of factor 10. Clear? So the pathway, the intrinsic pathway starts with activation of the factor 12, then factor 11, factor 9. Activated factor 9, factor 8 and calcium ion together work on inactive form of factor 10, and the factor 10 is converted to its active form. Now the extrinsic pathway. This pathway is a shorter one. It starts with factor 7 activation. Whenever there is any injury, tissue thromboplastin is released from the surrounding tissues. Tissue thromboplastin acts on factor 7 to activate it. So there is factor 7 which is converted to 7A by the action of tissue thromboplastin. Now the factor 7A along with tissue thromboplastin and calcium ion works on factor 10 to activate it. The intrinsic pathway and extrinsic pathways are done. Till now we have activated factor 10. Let's see what happens in the common pathway. Factor 10A joins with activated factor 5 and calcium ion to produce a complex prothrombin activator. What is the function of prothrombin activator? As its name suggests, it will activate the prothrombin to thrombin. Now this thrombin will act on fibrinogen. Can you remember, while discussing the components of blood, I have told you that fibrinogen is a clotting factor which is present in plasma. But this fibrinogen is water soluble. Can we stop bleeding with a water soluble substance? No, we can't. So anyhow, this water soluble fibrinogen has to be converted to insoluble form. Thrombin works on fibrinogen to produce insoluble fibrin monomers. So thrombin acts on fibrinogen which is soluble and produce fibrin monomers which are insoluble. Now these fibrin monomers polymerize. They polymerize to form fibrin meshwork. But this fibrin meshwork is soft. It is not so much powerful. Now factor 13 acts on it to make it firm and stable. Factor 13 is also known as the fibrin stabilizing factor. The formed elements of blood get trapped in this fibrin meshwork and ultimately there is formation of a clot. Now there is clot retraction with expulsion of serum. What is serum? Serum is the plasma without the clotting factors. It is a pale yellow colored fluid that comes out from a wound after the formation of a clot. What are the vitamin K dependent clotting factors? We have 4 clotting factors which are dependent on vitamin K for their activation. These are factor 2, 7, 9 and 10. Let us know the name of some anticoagulants. As vitamin K helps in coagulation, vitamin K antagonists can act as anticoagulant. We have warfarin, dicumarol. These are the vitamin K antagonists which are used as anticoagulants. Some other types of anticoagulants are heparin, EDTA or ethylene diamine tetraacetic acid, sodium citrate, double oxalate, etc. Now, what is bleeding time and what is clotting time? Bleeding time is the time taken for bleeding to stop. Normally it is 2 to 4 minutes. It depends on the formation of a platelet plug, and the clotting time is the time which is taken for clot formation. Normally it is 3 to 5 minutes. Now, bleeding time and clotting time can vary according to the depth of the wound, the site of the wound, and some other factors. This is all for today. If you found my video helpful, hit the like button, share it with your friends, and don't forget to subscribe to my channel. Press the bell icon to get notified about new videos. Thank you.